In this lecture, we'll be going over the development of the limbs. We'll start off at week 4. By the end of week 4, we can see tiny little outpocketings on the lateral body wall. So here's the upper limb, that little bump, and then the lower limb forming on that lateral body wall. It will have an ectoderm covering. Here's in blue is the ectoderm. This will form the skin of the upper and lower limbs. And then in red here is the mesenchyme. This will be a mesenchymal core. This will give rise to the bones, cartilage, and ligaments, as well as the muscles and nerves of the upper and lower limbs. The ectoderm will thicken at the distal tip, and this thickened region is called the apical ectodermal ridge. And I will just abbreviate it as AER. The function of the AER is it will induce the differentiation and the growth of the limb bud. It will tell the underlying mesenchyme to remain undifferentiated but to rapidly proliferate. And this region is called the progress zone. And essentially, the AER will tell the limb bud to grow longer and longer. So now we are on week 5 at that limb bud stage. Now the limb's starting to look a lot longer. Here's that mesenchymal core, the AER. As the AER and the progress zone move further and further away from the lateral body wall, as those cells move farther away from its influence, they will then condense and differentiate to form the cartilage and muscles. And for this example, we'll pick the upper limb. So I'll draw the humerus. That's a lovely humerus. And the radius and the ulna. So as this region is moving further and further away from the mesenchyme, it will start to differentiate into the cartilage, and the muscles will migrate in from the myotome, and their nerves, their corresponding nerves, will move in as well. Week 6, we will see the development of hand and foot plates. Let me draw that out here. The limb at the very distal part of the limb, it will flatten, forming hand and foot plates. Here's the humerus, radius, ulna, and then within that hand and foot plate, the digits will form. Those fingers will form. Got digit 4 and then digit 5 right here. And these cartilaginous digital rays, they'll be kind of trapped within this paddle-like structure until programmed cell death or apoptosis between each of the digits. And this cell death is what sculpts each of those digits out of this foot paddle or hand paddle. You might be wondering, well, how does the pinky and the thumb form? They're on different sides. There's what is called the zone of polarizing activity, or ZPA, that will form on the ulnar side or the pinky side of the limb. And essentially, ZPA will secrete morphogens, and it will create a concentration gradient across the length of the limb. So a higher concentration will be located near the ZPA and a lower concentration further from it. The high concentration near the ZPA will tell the underlying mesenchyme to make a pinky. The further away or the lower concentration of morphogens will tell the mesenchyme to make a thumb or to make the radius. And then by week 8, we have those fingers and toes fully formed. Between weeks 6 and 8, I'll put it in green here. Week 7, the limbs will actually rotate. The upper limb will rotate laterally 90 degrees, and the lower limb will rotate medially 90 degrees. And this is pretty much why the big toe is located on the medial aspect of the foot, and the thumb is located on the lateral aspect of the hand. In summary, limb formation begins in week 4 with the development of the AER. It will cause the outgrowth of the limb buds from the lateral body wall. Week 5, we had the limb bud stage. Week 6, we had the development of those hand and foot plates. And then by week 8, with apoptosis, those fingers will have been fully formed.